Oh, that's great. I'm good. Good to hear that you're pretty good. Um, so, okay. So today's lecture was designed to be a little bit more of an interactive lecture. So I'm going to definitely count on you guys who are here to put a response in the chat when you can. But today's lecture is on reading experiments, and I'm Jenelle. I've been here for a little bit at Socratic Med. Yep, everyone in okay. Yes, and this is just a reminder for those who are on this call right now that this lecture will be recorded and it will be posted to YouTube afterwards. So just a reminder to, change profile pictures, change your names that you are comfortable with to be public on, YouTube and the recording. And so we are, I believe, on week 13 of our 15-week full MCAT course, which started on June 17th. There have been 3 lectures per week from Thursday through Saturday, 8:30 p.m., and also an office hour session every Sunday at 8:30 p.m. Our aim was to cover the highest yield MCAT content during this period, and so you can look at all of the past recordings that we've had on our YouTube channel. And then also our GroupMe is here if you wanted to get in touch with the tutors, or also if there are any changes with the course or anything like that. Okay. So today, the learning objectives, I guess, are pretty simple. It's going to be a little different than the lectures that we usually, or the format of the lectures that I usually present. But today, mainly just to go over some tips to help with reading and comprehending experiments and graphs that are found in the MCAT science sections, because I've heard that students really wanted to improve in that aspect. And also, specifically today, the passages that we're going over are focused in the biochem section and the psych soc section. And so we're going to go over some practice passages today. So I'm going to start off with sharing just some tips of how to go about the passages. So I guess my tips for going through a passage is first to gather information. And so when you're looking at the passage, you want to notice familiar terms. So when you're looking at the passage, you want to notice familiar terms and things that you can really integrate to help understand what you're reading in the passage. So that's the first step that I usually focus on. And then the next tip is trying to understand the figures based on their reference to it in the passage. So usually when you read or in biochem or psych soc passages that have an experiment, you will notice that they will kind of go over the main gist of why they want to perform this kind of experiment, and then they'll go into what they're looking and then refer or make a reference to like figure 1. And so when you see that reference, you really want to look at that graph or other types of figure, because it could be a picture, it could be something else to kind of show like the structure. But you want to look specifically if it's a graph or like a picture that shows like a blot of some sort. I would say the main things first if you want to break that down, first identify the independent variable, which is usually on that x-axis, and then also identify the dependent variable on the y-axis. And when you look at the figures, I would suggest just explaining briefly, or at least comprehending what the figure is trying to say, and just do like a brief summary. And then after these two steps, I like to sometimes propose one thing that a researcher might want to do next, just because I feel like it helps me really understand what they've done so far and what could be improved. So those are my 3 tips. And so, right, we're just gonna go into practice passage 1. So I have the passage here, but I also am going to put a link in the chat with the um, link so that you guys can open it on Google Docs on your own. Let me just figure that out. Okay, so I just put the link in the chat. But I'm going to give you guys 10 minutes to read over this passage, and then we're going to regroup, briefly read it together, and then go over, questions. Okay, so 10 minutes has passed. Is everyone good, or do some people need a little more time? You can put it in the chat or unmute. Good. Okay. Anyone else? Just double checking. No one else needs more time. Oh, do you see it now? Okay. Okay, that's good. Thank you. Okay, so then we're gonna go through this together. I'm just gonna read through the passage first. So we see here, the passage reads, post-operative acute kidney injury, AKI, is a severe complication after liver transplantation, as its deterioration and magnification can lead to an increase in mortality. Connexin 43, abbreviated CX43, mediates direct transmission of intracellular signals between neighboring cells, always considered to be the potent biological basis of organ damage, deterioration, and magnification. So,
Tell me hello. He wouldn't understand. He thinks.
Sorry, could you just remember to mute, please? Thank you. Okay, so let's just first focus on the first paragraph. We see post-operative acute kidney injury. So that is the main, acute kidney injury is the main like term so far. And it's a severe complication after liver transplantation. So, you know, liver transplantation is associated with leading to acute kidney injury. And then we see that there is a special term, connexin 43, which is mediating that transmission of intracellular signals between just neighboring cells. So just signaling between cells. And also, you know, so now we're thinking about signaling to show this, to lead to the other cells knowing about this organ damage. So now we're going into the second paragraph. Researchers obtained donor liver tissues of patients in order to explore the effects of CX43 on organ damage. Reperfusion is a biological process used to refill blood in AKI. Donor livers displayed more severe lobular distortion with necrosis, apparent edema, hemorrhage, and neutrophil infiltration after reperfusion, compared with donor liver tissues obtained before reperfusion or normal liver tissue, for example, from patients with hepatic hemangioma. After reperfusion, CX43 mRNA and protein levels were determined as shown in Figure 1. The Figure 1 is here on this left side, and it says CX43 mRNA and protein levels in normal or donor livers before reperfusion and after reperfusion. So based on that second paragraph, we see specifically the researchers wanted to see how CX43 is associated with organ damage. And they decided to, you know, study how the different donor livers express CX43 mRNA either normally before the reperfusion or after the reperfusion. And so the main thing here so far is that donor livers displayed more severe, you know, all of these different problems after reperfusion. So this specifically after reperfusion, you see all of these effects. And then specifically in Figure 1, we see here that you can also see that through the CX43 mRNA units that seem to be increasing in here, the donor liver, and specifically after reperfusion. Also, something worth noting is that when it has these stars here on top for graphs, that usually means that there's a significant difference between this and the prior one for the most part. And that's where you can determine some conclusions if they give that as an answer choice. Could someone please mute? Okay, thank you. Does that mean it's statistically significant? Yeah, so the difference between like the after reperfusion and this before reperfusion is statistically significantly different. Like there's a difference. Yeah, so that's worth it to know when you see any stars like that on graphs, pay attention to, you know, what the effects are. Anyway, okay, so now we're done with the second paragraph. We're going into the third one, which says autologous orthotopic liver transplantation AOLT rats were built by researchers in order to further study AKI. Researchers hypothesized that the change in CX43 expression might play an important role in AKI following AOLT. Researchers used heptanol, a well-known inhibitor of CX43 without hepatotoxicity, to alter the function of junctions composed of CX43. Heptanol has no effects on CX43 expression in kidneys or livers. So we see now that they're using these AOLT rats for another study to further study the main AKI term that we learned in that first paragraph. And we see here that they specifically use heptanol, which inhibits CX43, except so it alters the function of junctions, but doesn't affect CX43 expression. So then we see here, and then if we keep going through the next part, it says heptanol or a sham control were injected intravenously into the rats, and the pathological score of kidney cells was determined to analyze disease progression. The results are shown in figure 2. And so this is where we lead into figure 2 here, which says the pathological score of AOLT mice treated with CX43 inhibitor, which we know based on the passage is heptanol. And so we see here the x-axis. So this is 8 hours after reperfusion. So we know that this is already like an after reperfusion state. And then they said earlier here a heptanol or a sham control, but we can see here that there's a sham, and then there's also a heptanol and sham option, and there's just the AOLT, and then there's also a AOLT and heptanol option. And we see again here are like asterisks showing the differences between the bar from the prior factor. And we see here that the pathological score, is higher as it goes up. And so we see that there are some differences between these groups, which we will probably go a little bit more in depth into a little later. So this right now is after figure 2. And then we go into here, the last statement that they make in this passage. Researchers also believe that RIP1 may be involved in CX43 function, and they confirm the interaction of the proteins through co-immunoprecipitation. RIP1 is a typical marker of necroptosis. They kind of leave it at that. So now we're going to kind of go into just reviewing the passage. So I just wanted to make sure. First, what do you guys feel are the main points of interest in this passage based on the first paragraph? You guys can put it in the chat or you can unmute. It's up to you. But just wanted to briefly review. Oh, Kazi?
Wait, you asked for the first paragraph, right?
Yeah, for the first paragraph.
Well, it emphasizes connexin's role in liver deterioration after surgery, after transplantation, sorry.
After transplant, yeah, so the CX43 is definitely something of interest, and let me just go back for the first, yeah, so the liver transplantation, as we can see here in the first paragraph, CX43. Does anyone else want to mention? Oh, I see, definitely the graphs. But just the note, the graphs come a little bit later. So specifically in the first paragraph, I guess, what are the main points of interest? Actually, I mean, Kazi definitely went over this. So I think we can go on to the next question, which is, what was the purpose of the first experiment, and when I say what was the result, I want to say, like, what were the main findings that you see? You can also put that in the chat or unmute. It's up to you. Experiment was to explore effects of connexin 43 on organ damage. Let's just so definitely, connexin 43 is important in that first graph, but focus on, it's not exactly connexin 43's effects, but remember, when we want to look at what is affecting what, we're looking at the x-axis for that independent variable that is affecting the y-axis. Yes, yeah. So definitely donor livers after reperfusion were seen to cause the most injuries, or I guess because I see, Kazi, you're going one step after what it shows directly in the graph, but that's definitely what you guys will, if if looking at graphs in these experiments are not as like easy for you, then this would probably be like the step after. But so we definitely know here, you see that the donor livers after reperfusion have this higher amount of CX43 mRNA units. And we know that based on the passage, CX43 mediates that transmission of signals that signals this organ damage and injuries. So, okay, good job so far. And then now I also wanted to know, what was the purpose of the second experiment and what were the main results? So if you want to go back to the second one here. Right, yeah, so what Kazi said was the result of the first experiment. So in the second paragraph, it goes over the first experiment where it says, they obtained these liver tissues, donor liver tissues, and then it said that these donor liver tissues displayed more, all of these, injuries after reperfusion compared with those that were before or before reperfusion or normal. And so we see here that there are the labels for this first bar being normal and then before and after. So yes, this is from the first experiment. So now for this question, we're looking at the second experiment. So anyone can just take a stab. This is definitely completely a learning environment. So I would say it's better to get out all of your thinking here before needing to, or before taking the exam for real. And like David said, yeah, so specifically in that first experiment, directly based on the graph, we're seeing that liver damage after, well, it's donor liver tissues after reperfusion is positively or is associated with the higher CX43 mRNA. Okay, so the second experiment is using peptinol to alter junctions made up of CX43 in rats. So okay, so definitely right in terms of they are using heptanol and they are affecting the junctions for some of them, like for some of these groups that they tested on, and this was in rats. But I guess based on the graph, do you see anything that stands out to you of maybe like a particular group that looks a little higher than the other ones? Just a reminder to, oh no, no one's talking right now. Yeah, we're just so just a reminder, the x-axis and the y-axis. So definitely just put out a guess. So
What did you ask? Sorry, I just rejoined.
oh yeah, no worries. I was asking about what were the main findings from the second experiment. So figure 2. And Fabliha was, I mean, she definitely went over that, you know, they are using heptanol to alter junctions in rats, right? But if we're looking at this figure, is there something that kind of stands out to you based on looking at all of these different groups and results from these groups that you see here? Okay, 1, the sham control has the lowest pathological score. Anyone else want to add something? AOLT has the highest score. Yep, definitely. And if we want to keep going on that, so how about the heptanol? What did it, because we know that they're using heptanol also for this. So how did it seemingly affect any of the groups? Heptanol makes a significant effect on PATH score on AOLT mice. Definitely, yes, yeah. So we see here on this last bar that adding the heptanol leads to that pathological score suddenly decreasing a lot more than before. Great. Yes, Kazi?
I'm confused about something. I don't know if I'm like reading it wrong. So I understand that heptanol inhibits connexin, but then in the last sentence of the third paragraph, it said it had no effects on connexin expression in the liver. So I found that contradictory. I'm not sure if I'm like understanding it wrong.
Right. So, so when it says it affects so it's not exactly contradictory. So, it can alter the function of the junctions, meaning that that signal is not, because we know here in that first paragraph that CX43 mediates that transmission of the signals between neighboring cells, which would most likely be through junctions of a sort. And so this heptanol that inhibits it affects those junctions, so not allowing it to really like pass through or send those signals. But then it doesn't have an effect on CX43 expression. So meaning that if the kidney cells or the liver cells are still, producing the CX43 from their direct, like that cell that's directly doing that, the expression is still there. It's just not quite, getting sent over to the neighboring cell.
Oh, okay. I see now. Thank you.
Of course, yeah. Okay, yes. So great job, guys, so far. And then this is just an extra question. What is another experiment that the author could do based on this? I would say when you're thinking of another experiment, what helps for me usually is looking at what happens at the end, or if they might say an additional statement at the end. So if it helps, I'll move back to the last one. Yes. The RIP1 thing. You guys, yeah. So could you like elaborate a little bit on that?
This is a guess. This is actually the first passage I'm doing ever.
Oh, don't worry, yeah.
But
Don't
I guess
worry.
what I would, from what I don't know what necroptosis is, so I'm not even going to comment on that. But since they believe that they might be involved, they could set a sort of like a similar experiment to figure 2 in which they can treat the transplanted liver with RIP1 and then another one with no RIP1 and see if it has any effects, indirect or direct, on connexin.
So with or without RIP1 and how it could affect connexin.
Yeah, how could it, yeah.
Okay, yeah. So definitely that's definitely an experiment that they could do next. Does anyone else want to give another thing, or should we move on to the first question? What do you guys want to do? Move on. Okay, so we're gonna go to the first question. So just first question is, what type of junction does CX43 most likely form? So I'm gonna put up a poll, and then we're gonna, I'm gonna give you guys a minute and let me know what you guys think. You can also refer back to the passage if you want to, like, check on that doc. Okay, I'm gonna give you guys 15 more seconds if you guys want to get your answers in. Okay, so we have 67% saying A and 33% saying I have no idea. Okay, so I would say the majority of you guys, great job, because it is A. What type of junction would CX43 most likely form? It would most likely be the gap junctions. And we can go into that right now. So in that first paragraph, we see that CX43 mediates direct transmission of intracellular signals between neighboring cells. And so they're transmitting these signals between neighboring cells, and it's that's like what type of junction we're basing this off of. So I would also say that they like to ask this kind of question pretty often on the exam. And so it's definitely worth it to know the differences between gap, tight, desmosome, not as much hemidesmosome, but the first three definitely. So I'm going to show you guys a figure to help with just, visualizing the different junctions. And so the reason why the answer is gap junction is because these cells are transmitting these signals between the neighboring cells. And gap junctions are one of the few that allow cell signaling to be passed through their junctions. And that's just something that you guys will have to know for the exam. And so that's gap junctions, right? So then we're going into tight junctions. So what tight junctions are is it connects the two membranes of the neighboring cells so that there isn't anything that's particularly leaking out from the cells. And so if we're thinking of, that CX43 like we had in the question, this CX43 would not really be able to pass through as much. It would kind of just be stuck because usually signaling cells are a little bit bigger, and tight junctions really make sure that the the fluids within the cells kind of stay inside, and maybe some small like ions, but not really a signaling molecule it wouldn't really pass through a tight junction. And then desmosomes, on the other hand, they actually just so they're linked to the intermediate filaments inside the cell, and they allow these cells to be like stuck together in a sense. And so desmosomes kind of act as like a connector between the two cells that just like let it, stay together. And then hemidesmosomes, they're linked to intermediate filaments, but to the extracellular matrix. So this is making these cells stuck to the extracellular matrix. And then so that's why gap junctions are the correct answer. Also, specifically in that first paragraph, they mention like that the signals are sent between intracellular, they're intracellular signals. And so that if we're also thinking of like in terms of tight junctions, they focus more on they just connect at the cell membrane. So they're not really going into that, they're not really passing through any intracellular material over to the other side. Okay. So great. If does anyone have any questions about this? Okay, if not, we're gonna go into the next question, which I will also give you guys a minute about to think about. Okay, I'm gonna give you guys 10 more seconds if you guys can get an answer in. Remember, this is all anonymous, so completely fine. Just put whatever you think that the answer is. And we're definitely going over it, all of these. So, yeah, take your best educated guess. Okay, ending poll now. Okay, so I see we have a split here. So we have 33% saying B, 33% saying C, and 33% saying I have no idea. Okay, so let's just read the question first. A researcher discovers a new CX43 inhibitor made of a glycerol backbone and 3 saturated fatty acetyls. Which of the following describes the path by which the inhibitor enters the cells? So something to note about, I would say, biochem passages and chemphys passages is that in the questions in particular, they will sometimes give hints or give properties that already hint towards what may be the answer. So we see here that this inhibitor, this new one, is made of a glycerol backbone and 3 saturated fatty acetyls. Now, before we go into the answer, what does a glycerol backbone and saturated fatty acetyls kind of remind you of? Put in the chat, or lipid, hydrophobic. Right, exactly. So like if we're thinking even just like the cell membrane, the phospholipid bilayer, that, I mean, it's the structure is a little different, but like with a glycerol backbone and one saturated and one unsaturated, but definitely like is mentioned in the chat, lipid hydrophobic, right? Because of these saturated fatty acetyls, and there's all this like hydrophobic aspect to it. So then that kind of gives a hint. So the answer is C, with the inhibitor diffusing through the cell membrane, because we know that the cell membrane regulates what goes in and out of the cell. And a lot of times, so this is just also something to know, definitely something that's important to know on the exam, are the properties of the different things that make up a cell, of a cell. So like the cell membrane and what molecules can easily, pass through a cell versus what things cannot pass through that cell membrane. And we know based on past information, or if we remember the lecture that went over the cell membrane, that very hydrophobic molecules easily pass through that cell membrane through that phospholipid bilayer. And that's why C is the correct answer for here. In terms of the inhibitor being recognized by a specific receptor or taken up by a non-specific receptor, and also passing through the cell membrane via a channel, usually those things would happen in a typical signaling molecule, which tends to be charged. And so that's why, based on what this new inhibitor's properties are, we can narrow down this answer choice to be it diffusing through the cell membrane. Does anyone have any questions about this one? Okay. In that case, I'm going to go on to the next question. And I'm going to launch the poll again. So this is a little bit of, thinking about, kind of what I mentioned about thinking of a new experiment that someone could do. Okay, so 10 more seconds, guys. If you can get your final answers in. I'm ending the poll in 3, 2, 1. Okay. So we have 67% saying A and 33% saying I have no idea. So I will say the majority of you guys are right. So congrats. So the question says researchers decide to interrupt the CX43-RIP1 interaction in the AOLT rat model without using a small molecule inhibitor. Which of the following will most likely disrupt the interaction? So we're focused on the interaction between CX43 and RIP1 that we know based on the passage seems to have an association with each other. So now we're going to look into the answer choices. So let's start off with B, radiation against heptanol. So we know that heptanol is an inhibitor of CX43. So if we were to do radiation against heptanol, we would kind of then get rid of the inhibitor for that, which wouldn't exactly cause a disruption in the interaction. And I feel like radiation also is pretty general because you would most likely end up just putting radiation onto the cell, which then would kind of interrupt just entirely like the cell's processes. So that might not exactly be the best one to focus on the interaction between CX43 and RIP1. So then next we look at C, acetyl transferase specific for beta actin. This is also not quite related to what like the interaction between CX43 and RIP1 is. So based on the figure 1, if you remember from the passage, there is a part underneath the graph that shows the protein levels or like the protein bands of what appears there, and that's where it mentions beta actin, but beta actin is not directly responsible for the CX43 and RIP1 interaction. So that's why C is not going to be the best answer. And then D, plasmid producing CX43. So with the plasmid, it's producing more CX43. So if we're thinking if the rat model can take up this plasmid, it would end up kind of increasing the interaction because then it wouldn't really be disrupting the interaction. You would have more CX43, and then if there is RIP1 that's excess, then that CX43 could interact with the RIP1. And so that's why C is not quite disrupting the interaction either. So this answer choice, siRNA for RIP1, is a little bit based on more, I guess, background knowledge about siRNAs. And I would definitely suggest knowing these different inhibition methods, or like this definitely you should know. And also a lot of the, I would say, is it like molecular processes? That's very important because they do like to use, they definitely do have a question for signaling molecules. I would say at least one passage in every exam. Signaling is very heavily tested on, so definitely understand the process of signaling molecules and different types and what can cause that transmission to not really go through. And so with siRNAs, they bind to like the mRNA. So we know with CX43, it's producing that CX43 mRNA. So it would bind to that and effectively just degrade the CX43 mRNA. And so when you're degrading that, then it would most likely disrupt this interaction, because then RAP1 wouldn't have CX43 to bind to. So does anyone have any questions about this one? You can put in chat or unmute. Up to you. Okay. So if not, then I have one more question left, I believe, for this passage. And I'm going to turn up the poll again and also give you guys a minute. Okay, so I'm going to give you guys like 15 more seconds. We definitely went over this one together, figure one, if you guys remember. So definitely put in an answer, even if it's a guess, into the poll, if you can. Okay, ending poll in 3, 2, 1. Okay, so we have 67% saying C and 33% saying I have no idea. So definitely you guys, the majority got it correct. The answer is C. So this question is just, focusing on which conclusion about CX43 regulation is best supported by figure 1. And so if you remember our discussion about figure 1, I was trying to, get from you guys what stood out to you most in that figure. And someone mentioned definitely that, choice C, donor livers have higher levels of CX43 after reperfusion compared to a normal liver or even a liver before reperfusion. So that's why C is the correct answer. If you have, so with choices A and B, so that's all expression control occurs at the transcriptional level and all expression control occurs at the translational level. So this, you can see in figure, here, I will go back to practice passage 1, but see, so this is figure 1. So it includes both E and G, and with E, that's focused on the mRNA units. So we see, and also these groups correlate with the ones below that. So the normal liver tissue right here don't really have a band of CX43 showing, but so with the first answer choice that was saying that it was all based on mRNA, that based on this figure that we're seeing, the mRNA levels are increasing here, but when we're looking at this expression of CX43 on that protein level, we're seeing that the first two groups don't have any protein expression, and then that last one has some kind of expression for, we are not sure why that's the case. And so when we're looking at this these answer choices saying that all expression control occurs at the transcriptional level and at the translational level. We're not entirely sure of that based on that figure, right? Because we're suddenly seeing that the mRNA levels are a little different. And also like the proteins that actually get produced at the end are different. So we don't know which step exactly is really controlling that expression at either level. And so that's why for A and B, those would not be able to be concluded from figure 1. And then for choice D, it says normal livers treated with heptanol have lower levels of CX43 compared to donor livers treated with heptanol. So if you guys remember, heptanol, that was definitely focused on in figure 2, not figure 1, and figure 1 doesn't show that information, and so that's why choice D is incorrect. So I believe after this question, we're going into the next passage. So does anyone have any questions about this passage in general, or want to just talk about their experience going through the MCAT passages in general? Like, did some of the tips help you guys for this first passage? So I'm not entirely sure of the difficulty level of this passage. Also, if you guys might have seen from the document, this one was from the Shenmisen. They posted some example passages for like the biochem, or I think it was just the bio section, and this was from there. So it's not exactly a, you know, AMC passage, just because I think that we aren't allowed to share that. But this is just like a general format, and I would say that this passage is pretty similar to kinds that you might see. Okay, so online, yeah, don't overthink. Some can be tricky if the experiments can't be kept clear. Right. So definitely when you're looking at a passage, so that's why for the first tip, I definitely say look at familiar terms and really distill the information that you're given, because the information in the passages can definitely seem very overwhelming, and you are not expected to know every single term that they have in that passage. It's really, they want you to apply that foundational knowledge that you know and that you guys should know really well by the time that you're taking your exam, and apply it to all these different scenarios that they might give you. So like with the gap junction question, they specifically say in the passage, like these signals are being sent intracellularly between neighboring cells. And so when you read that, you might be like, oh, I feel like that's gap junction. So they're being like transported between gap junctions. And then you kind of just like piece together the things that you know with what is given in the passage, and those like statements you can definitely say for yourself as you're going through the passage. And then, yeah, so the experiments, when you're, again, reading all these complex terms, it becomes really hard sometimes to feel like you understand everything that they're saying. But just, focus first on that X and Y axis and just see what the effects are on each other. Because that's the baseline. You don't even have to connect it to the bigger picture yet. Just focus on that graph and just see, because definitely in this question that I have up here, right, it says the conclusion from figure 1, and it's something as simple as, you see that there are the higher levels of CX43 mRNA after reperfusion when compared to normal liver tissue. So I would say definitely with passages that give you an experiment or figures, they are very likely to ask about the figures, but you also can be pretty, it doesn't have to go so like out of scope as to what usually you might be able to get from the figures. So definitely something that, oh, let's see. Whenever we do a passage, should we do what you did, like break down, oops, sorry, should we break down main idea of each paragraph and then the graphs? So I definitely feel like this is up to your different styles that you might develop as you're going through passages. But for me personally, what I felt helped me was to break down like the main idea of these passage chunks and the graphs to just, distill the information, be able to see how this figure and the results from this figure connects with that main idea and what like the researchers are trying to get from the experiments that they're doing. And so I definitely feel like it helps me visualize what's going on in the experiment. So that's why I give these tips that are, I guess, more of like my personal tips, but if it helps you, I definitely feel like you can do that. And like David says, yeah. So yeah, like David says and what I said, the passages always look scary because there's just all these terms that you may never have seen before. But focusing on the information that you know. Also, he says here like the abbreviations, italics, negation words, and the graphs definitely look at that stuff. And also, I know some people suggest roadmaps for cars, like jotting a sentence about the main idea. Should we do the same for science passages, or is that too time consuming? So kind of again, it really depends on if you feel like it helps you when you're doing these science passages to do that method. Because definitely different techniques work better for different people, and like it really depends on the person. And I think especially if you are trying out these passages and just continuing to practice, try it out a little bit and see like how it helps you understand the passage. If it definitely helps, then I would suggest, continuing to do that, because then you are getting into the habit of like doing these things, and it really does help you understand the passage to answer the questions. But if you feel like it is not really helping you that much and you kind of find a different way that helps better, then I would suggest doing that other way, because it is what works best for you in that case. But if you do already do roadmaps for cars, I mean, then I would say, like, if you do roadmaps for cars and it helps you, then I would say probably it might help you with science passages too. Seems like it would. But definitely try all these methods out during practice. But no matter like what method you use, definitely focus on those terms that you know, and don't get overwhelmed by all of the terms that they kind of throw at you on the passage, and focus on the graphs, because regardless of if you do the main ideas or not, those graphs are going to be definitely important. So you definitely have to understand those graphs and figures that they give you. Okay, so that was with the biochem passage, or biochem types of passages. And so as you can see, based on the one that we did, it was very like, there are a lot of layers to what we just read, and also even like the questions that they ask, like with that lipid, with that inhibitor question, with that hydrophobic inhibitor question. So you will see often that the biochem passages and the chem phys passages will be pretty intense with connecting all of these different things that you've learned and applying it to all these different situations. So now the next passage that we're going to do is a psych soc passage. And what I feel like is a little bit different about the psych soc passages is that they tend to be a little bit more simple in the sense that they don't usually have all of that complicated jargon or really kind of really complicated terms that you might not have heard before. So for psych soc, I feel like it's pretty common terms. And I feel like if you know all of the psychology, sociology terms that you should for the MCAT, I think that it's relatively easier sometimes than the biochem / chem phys section. So without further ado, this is the second practice passage, and we're gonna give you guys, I'm gonna give you guys 7 minutes, and then let me know how it goes, or if you guys need more time. Okay. How is everyone feeling? Is everyone okay and done with reading the passage? And just put a Y or N in the chat. Yes or no? Yes. Okay, great. Okay, then we can start going into the passage together. So the passage says, recent studies have shown that television can adversely impact cognitive processes in both children and young adults. In previous studies, researchers found that individuals ages 6 to 23 higher than average amounts of television watching led to an 8 to 14% decrease in the ability of participants to quickly and correctly solve math problems. In addition, participants were less likely to recall list items or accurately identify emotions on a confederate during an in-person experiment. So we see here, based on the first paragraph, recent studies show that television can, cause 8 to 14% decrease in the ability of participants to quickly and correctly solve math problems, or just, impacts their cognitive processes. So that's mainly that first paragraph. And then the second paragraph, a group of researchers is specifically interested in studying the effect of television on memory. The researchers recruit at least 50 participants in each of the following age groups: 6 to 8, 9 to 11, 12 to 14, and 15 to 17. Each of the participants watches 15 hours of TV per week as confirmed by the participants themselves and a guardian. The participants watch a range of television programming throughout the week. So in the second paragraph, it's setting up the experiment to say that the researchers tested different age groups, and each of these groups watched 15 hours of TV with a range of different programs, but just that they're watching that 15 hours of TV per week. And then for this third paragraph, to conduct the experiment, the researchers split up the participants in each age group into group 1 or group 2. Group 1 participants watch 15 hours of TV a week for 3 consecutive weeks. And at the end of the third week, the participants are brought into a testing center and given a list of 20 names. 10 minutes later, they are asked to recall as many names as possible. The group 1 participants then watch 15 hours of TV a week for the next 3 consecutive weeks, weeks 4 to 6, and repeat the memory assessment. Group 2 participants follow an identical plan as group 1 participants for week 1 through 3. However, during weeks 4 through 6, group 2 participants are not allowed to watch any TV during the week. Group 2 participants complete the same memory test as Group 1 students at the end of week 3 and at the end of week 6. The results from the experiment are shown in Figure 1 for the 9 to 11-year-old age group. And we see here, this is the figure for the 9 to 11-year-old age group. So based on this third paragraph, we notice that the researchers are splitting the participants into Group 1 and Group 2. And Group 1 and Group 2 differ just in the week 3 to 6 area, where for Group 1, in weeks 3 to 6, the participants are still watching 15 hours of TV a week. But for Group 2, in weeks 3 to 6, they are not watching any more TV during the week, and they're still tested at the same time points. So we see in Figure 1 here that there is on the x-axis a group for the week 3 test, and there is also a group for the week 6 test. So we see also the number of names recalled. And based on this, it also has the color coding for Group 1 and Group 2, and we're seeing how the effects of being in these different groups, and also when they're tested at the week 3 or week 6 stage, affects the number of names they could recall. So kind of going over what we did last time with the passage. What was the main point that they wanted to mention in that first paragraph? You can just put it in the chat or unmute, up to you. Just a simple, like few words probably would be able to sum up that. Okay, how about okay. Yes, definitely. Yes, exactly. So television lowers the cognitive abilities of individuals. And that affects memory and all these other things that they're testing. So great. And how about this passage only has one figure. So what is something that you're noticing based on the figure? Group 2 in week 6 test recalled more and nothing is statistically significant. Yeah, so both of you, oh, hold on, wait. Reading the chat. Okay. Yeah, so we see here definitely both of you guys got points that you can see in this graph. So group 2 in week 6 recalled more, as we can see here. So that may be an effect by the difference between the groups. We don't know exactly, but like also here that nothing is statistically significant. That's also true because we don't see any stars or asterisks. So we do not exactly know that, there is this major statistically significant difference between this group 2 and group 1 in that week 6. So we see that there is like a higher amount of names recalled in group 2 at week 6 compared to group 1. It's just we can't exactly say that what we changed caused that. And I guess what is something that, potentially the researchers could look into next? You can completely take a random, I guess, angle on it. Yeah. So what is potentially another experiment that the researchers could do based on what they found from the first figure and the experiment that they did? So test it on a different age group. That I would say, I mean, they could definitely test it on a different age group. Probably like when you say different age group, greater than 17, probably, because that's, yeah. Okay, yeah. That's definitely, that's definitely an experiment that they can do. So great. Yeah, it's true. Thinking caps on. But anyway, so yeah, so that's definitely something that to do. I'm just gonna skip this slide because this is that, and then we're gonna go into that first question. So I'm gonna launch the poll and just put your best guess. I'm gonna give you guys a minute. Anyone want to get a last answer in, or just you know, take a stab at it? You could also pick I have no idea. Definitely an option. I'm gonna give you guys like 5 more seconds. Okay, so we are ending poll right now. We have 50% saying A and 50% saying C. So 50% of you are right. But I would say it's a good thing. So the answer is A. It's a good thing that you guys didn't pick B or D because those are a little bit different. But and at least, you know, both the primacy and recency effect are kind of like under the same like set of ideas. And so I'm going to explain this after going through this question. So it says, in a follow-up study, researchers found that participants are best able to recall names at the end of the list better than names at the beginning of the list. And so this is known as, and it's known as A, the recency effect, because what the recency effect states is that when people are given a list, they tend to remember the most recent things that they read. So that would be like the last thing on their list. And so that's why A is the correct answer, because we can see in this question, they're able to recall names at the end of the list better than the beginning. And so on the other side, the primacy effect is when they believe that people can remember things better at the beginning of the list, or the ones that they're seeing like first. And so that is C. That's what, you know, the term in C means. And so with B and D, so proactive interference and retroactive interference are when, so proactive interference is when the past things that you have remembered in your brain kind of affect the new things that you're trying to learn. And then with retroactive interference, it's when new things kind of affect the things that you have already learned in the past. So I guess the way I kind of help to remember it, because I know when I was studying, I used to get a little mixed up between the two. I think retro as in like the past, and the retro is the one that's interfering with, you know, what you're trying to learn. And then with the proactive is interfering with, you know, what you already know. So does that make sense to everyone? Okay, so I think we're gonna go into the next question. And I'm gonna give you guys like a minute as well. Okay, like 5 more seconds. Okay, and then poll. So we have 50% saying A and 50% saying C for this one. So the answer is C. So the question asks, which of the following do the researchers not account for in the study? So A says intergroup variability. And so in a sense, they do account for this when, if you let me refer to the passage. So we see here that the group of researchers recruit 50 participants in each of the following age groups. And so they are aware that, each of these age groups may have differences. And so they did the same group 1 and group 2 on all of these different groups. All of these different age groups. And so they do, I'm sure, although they only showed figure 1 for the 9 to 11-year-old age group, I'm sure that they also noted the differences between these different age groups. And so that's why for A, that's why that's incorrect, is because they do account for this, and that's why they did all these different age groups. And so that also goes into why D is not the right answer, because both of them are different. And then for B, amount of TV watched, they do account for this in the study, because they specifically say for group 1, they have the 15 hours per week. And for group 2, they also have group 2 and group 1 differ in that 15 hours between week 3 and week 6. So the answer is C, because they say in the question, sorry, not in the question, but in the passage, that they didn't specify as to what type of TV programs they watched. If you remember from here. So it says the participants watch a range of television programming throughout the week. We only know that they do the 15 hours, not necessarily the type. And what is intergroup variability? So intergroup variability is just differences between groups. But so when you think inter, like between. Even when you're thinking with intercellular, so between the cells. Or intermolecular, between the molecules. But anyway, so hopefully that helps explain what intergroup variability is. And yeah, like David said, inter versus intra is very helpful when you're also learning other terms. So inter is between different groups. Like I said, and an intra is within that same group. Okay. So now we're going to go into this next question, and I'm going to put up the poll. Oop. I'm going to give you guys a minute. Okay, so 5 more seconds and I'm going to end the poll. Okay, so we have 100% saying choice B. And so you guys, great job. So yeah, the question says, which of the following conclusions is not supported by the results shown in figure 1? And we know from our understanding of figure 1 that group 2 and group 1 showed that difference in that week 6 area. And they were pretty much the same at that first part. So for choice A, group 1 and 2 participants. Sorry, could you mute, please? Thank you. So with choice A, group 1 and 2 participants recall a similar number of names when they watch 15 hours of TV a week. So we definitely know that they do based on that week 3 group where they are doing that check-in when they watch that same 15 hours of TV per week, they recall the same amount of names. And for choice C, the ability of group 2 participants to recall names improved on the week 6 test. So we definitely see that there was an improvement. To what extent that improvement is, we don't know if there's a significant difference, but we can say that, you know, there was that improvement. And then for D, the number of names recalled by participants is associated with time spent watching TV. And this is something that we can make these associations. We just can't make like definite causal relationships. And so we know that watching less TV based on that group 2 is associated with, you know, better ability to recall the amount of names. And so that's why choice D is incorrect. For choice B, says retroactive interference prevents group 1 participants from performing as well as group 2 participants on the week 6 test. So based on that figure, we do not know at all what is causing group 1 participants from doing as well on week 6. And we definitely don't know if it's retroactive interference because there wasn't anything that was mentioned for that. And so that's why choice B is the correct answer, because it's not a conclusion that we can make. And side note, that choice B, I feel like we would probably assume or think that the group 1 participants performed not as well in week 6 because of the amount of TV hours watched, but that's like just another association. And also, I think I may have, so I'm just going to reiterate the difference between proactive interference and retroactive interference because I might have made a slight mistake when I was explaining it before, but retroactive interference is when, you know, so think of it as the retro interfered to the past information that you have in your head that was earlier on is interfered by new information. And with proactive interference, it's when the new stuff that you're trying to learn is interfered by, or the new stuff is interfered by the information that you have. Oh, did it sound similar, guys? Or did I make a slight mistake on that again? See, this is why also I feel like that term kind of was very confusing for me when I was studying. But retro from the past. So later learning affects earlier learning. So any new stuff that you're doing affects the stuff that you had before, and proactive, the stuff that you had before, affects any new stuff that you're trying. Okay. So now we're going to go into the next question. I'm just going to pull again. We only have a few more, so hopefully these questions are also a lot easier than yeah, a minute. Okay, guys, like 10 more seconds. And closing poll in 3, 2, 1. Okay, so we have 50% saying A and 50% saying B. And so the answer is B, experimental study. And the question just asks, which of the following best describes the type of study described in the passage? And so the reason why the answer is B is because we see that the researchers are manipulating their groups in terms of adjusting the amount of time that they're watching TV. And so they're trying to find that they're trying to control for a lot of things. However, based on, the experiment, they're not controlling for everything, but it is experimental in that sense. There is also a difference between like a controlled experiment and just like an experimental study. And so that's why, because the researchers are manipulating variables, that's why it's choice B. For A, an observational study, that's more in the sense of if you were to watch groups of those different age groups and just seeing like the amount of TV watched and looking at maybe their memory recall, but not quite specifically saying that they have to watch that 15 hours a week and splitting them in group 1 and 2. You're just kind of watching and not really making any other changes to their habits. For a survey study, that, as we know in this particular passage, they didn't really ask for any surveys. And for a case study, we're also not differentiating between cases and controls in this one. So that's why choice C and D are incorrect. Okay, so if anyone doesn't have any questions about this one, then we're going to go into the next. Oh, wait, sorry, that was the last question. Okay, so thank you guys so much for tuning in tonight. Really appreciate the participation. Really great. And so just a reminder that we have office hours every Sunday, 8:30 to 10:30 p.m. EST. If you have any questions about this or any topics, I'm going to try to get this PowerPoint out to you by tonight, and the recording would be uploaded by YouTube tomorrow. So thank you.